Good afternoon, ladies and gentlemen. Let me first introduce my topic, that is the effect of KCS1 deletion on the therapeutic action of valproate. Valproate, valproate is a drug which has been highly prescribed to treat bipolar disorder, which is a very common psychiatric illness. And KCS1 is a gene, so my study will explain how a loss of a gene would affect the therapeutic action of valproate. As my study will involve with the bipolar disorder, let me briefly explain what is it. You might have familiar with this disorder. It's a chronic psychiatric illness and it's a brain disorder. Nearly 1% of the world population are victims of this disorder. In Asia, it's little bit more. I found an article, a review, it's 1.4% in Asia. The patients with bipolar disorder undergo unusual shifts in their mood, energy, as well as their activity levels. Therefore, they lose their relationships and they perform poor job and school performances. It has been found 15% of the bipolar disorder patients commit to suicide. Let me briefly go through the symptoms. The patients with bipolar disorder have extreme mood shifts. One end, which is called the joyful mood, called the manic episode, where the moods are elevated and they would like to involve in the activities more and more. And also they have an unrealistic belief in their abilities. Moving to the other end, the extreme end, where the moods are depressed, we call it depressive episode, and they lose their interest on activities. And they also think they are worthless. But there might be a mix-up of these episodes. But my study will not talk about anything with bipolar disorder, but the therapeutic action of the drug. So, what are the current treatments? Lithium, anticonvulsants such as valproic acid and carbamazepine, also antipsychotics are used. But among them, lithium and valproic acid are widely prescribed. But problem with this current drug is that they are not very effective, and 20 to 50% of the patients who have been treated with these drugs have failed to respond satisfactorily. And these drugs induce numerous side effects. So it is important to develop more effective drugs with less side effects. But the problem is the development of new drugs has been hampered by the lack of knowledge about the therapeutic mechanism of action of these drugs. Now, what is the therapeutic mechanism of action of lithium and valproic acid? To explain this, Berich et al. In 1982 postulated a hypothesis called inositol depletion hypothesis. That is, in the inositol biosynthesis pathway in the cells, glucose 6-phosphate is converted to 1D myoinositol 3-phosphate by myoinositol phosphate synthesis. This is the enzyme, and INO1 is the gene which codes for the enzyme. In turn, 1D myoinositol 3-phosphate is dephosphorylated to inositol. This is how inositol is synthesized in the cells. It's a dephosphorylation, so the enzyme is inositol monophosphatase. And INM1 is the gene that code for the IMPase. Further, inositol is further phosphorylated to different phosphatidyl inositol and inositol phosphate. Now, lithium directly inhibits inositol monophosphate. If this enzyme is inhibited, the conversion of 1D myoinositol 3-phosphate to inositol is totally blocked. So inositol levels go down. And valproic acid, it indirectly inhibits MIPS. If it indirectly inhibits, that also depletes the inositol level. This is the entire pathway, starting from glucose 6-phosphate, 1D-myostol 3-phosphate, inositol, further conversion to different types of inositol phosphates. And these are the genes involved in this pathway. How this inositol synthesis is controlled? Inositol synthesis is controlled by regulation of this gene, INO1 gene. Let me explain how INO1 is regulated. Now, this is the upstream activating sequence of INO1 gene. N1, N2 are nucleosomes. To transcribe this INO1 gene, there are two activators called INO2 and INO4. These activators should be bound to the upstream activating sequence. But in the presence of inositol, these activators are unable to bind to this sequence because there is an inhibitor which blocks their way. So INO1 is not transcribed. INO80 is a chromatin remodeling complex. The role of a chromatin remodeling complex is whenever there is a gene is needed to be transcribed, it can rearrange the chromatin and it facilitates the binding of enzymes. In the absence of inositol, this inhibitor detached from the upstream activating sequence and facilitates the binding of activators to the sequence. Then chromatin remodeling complex INO80 stretch the DNA sequence, and on top of this RNA polymerase is bound and INO1 is transcribed. It has been also found that inositol 6-phosphate can inhibit INO80 complex, and in turn it can inhibit the INO1 expression. Now, what is IP6? IP6 in the inositol biosynthesis pathway, at the very downstream end, it is synthesized from phosphorylation of IP5 by IPK1 gene, and it is further phosphorylated to IP7 by KCS1 gene. My hypothesis. What I hypothesis was, by disrupting the inositol biosynthesis pathway, that can affect the inositol synthesis. Now, to do this, the model organism I used was yeast. You might be wondering how yeast can be used to identify biochemical pathways. Yeast is a eukaryote where the functional conservation is higher among yeast and human. And also, people have been widely used yeast to describe the action of valproic acid. And yeast is a microorganism where we can easily manipulate the genetic pathways. So, to disturb the biochemical pathway in the inositol phosphate, I have to delete gene by gene. But I did not wanted to delete because there is a deletion mutant collection in the yeast genome, where there are 4,500 genes have been deleted, and it is collected as a deletion mutant collection. So I pick those genes of the deletion mutants and grow them in the absence and presence of inositol. So this will show these are the different types of mutants in the inositol biosynthesis pathway. And I grew them. The one at the left column is in the normal media. And this one is without the inositol and with inositol. So you can see 3 of them did not grow in the absence of inositol. But these 2, VAC7 and VAC14, these are mutants in the vacuole. They even did not grow on the normal medium. So there was a problem with those 2 mutants itself. But KCS1, it grew without any problem on the YPD, but did not grow in the absence of inositol. But the growth was